William Ford's research paper, Fatal Pasteurella hemolytica pneumonia in bighorn sheep after direct contact with clinically normal domestic sheep, is presented by Ashley Hobbs, Caitlin Connor, and Katie Edwards. Here is some background for why the study occurred. Many bighorn sheep had died the previous winter in Wallowa County in Oregon. What the researchers already knew was that lungworm, pulmonary viral and bacterial infections increase the risk for respiratory tract disease, and that domestic sheep are carriers for P. hemolytica, and as well that bighorn sheep are highly susceptible to bacterial pneumonia and it can be fatal. The main object for this study was to evaluate the impact of respiratory tract disease susceptibility on the bighorn sheep that had controlled contact with domestic sheep.
To begin this experiment, the researchers got 5 Rocky Mountain bighorn lambs. They were born and raised at Washington State University. An additional male lamb was taken from the wild later on and included in the study. All of the sheep were held in captivity for over 1 year and were all clinically normal before the start of the research. 4 of the sheep were vaccinated subcutaneously 3 times. Each shot was administered 4 weeks apart and contained 2 milliliters of killed Pasteurella organisms. 6 domestic sheep, all clinically healthy, were introduced to the bighorn sheep. 48 days into the trial, the domestic sheep were removed. 3 were euthanized. Blood tests and fecal samples were taken the day the trial began and at the necropsy following the trial. They were evaluated for antibodies of viral pathogens in the respiratory tract.
Domestic sheep chosen for this study showed they were all initially positive for RSV, parainfluenza, and bovine viral disease. They were all negative for infectious bovine rhinotracheitis virus. The 6 bighorn sheep used in the study were all negative for parainfluenza virus, infectious bovine rhinotracheitis, and bovine viral diarrhea. RSV antibodies were found in 5 out of the 6 bighorn sheep due to the vaccination used in the study. Despite this, all 6 bighorn sheep died. Out of those 6, 4 were vaccinated with killed Pasteurella organisms and 2 were not. The 4 vaccinated sheep died on days 4, 27, 29, and 71. The 2 unvaccinated sheep died on days 27 and 36. When a necropsy was performed on all 6 sheep, it showed that lesions were present on the lung, lymph nodes, and heart. There was also significant necrosis of the lymph nodes, alveolar epithelium, and bronchiolar epithelium, as well as the lungs. Shown in the top right-hand corner is a sample of P. hemolytica under culture. This graph we made shows the progression of death every 10 days in the bighorn sheep in the days following the introduction of the domestic sheep to the herd of bighorn sheep. By day 71, all 6 sheep were deceased. This table was included in the original research paper. It summarizes the results of each sheep, both domestic and bighorn, that participated in the study. Note that bighorn lambs 5 and 3 were unvaccinated. Vaccination is indicated by a star next to the sheep's number in this table. This table shows for each sheep where P. hemolytica cells were isolated in the necropsy.
For the discussion section of the paper, the main points were that the contraction of pneumonia in the bighorn sheep shows that the vaccine that was administered to them before the trial began was ineffective, and that the bighorn sheep can develop acute pasteurellosis after coming in contact with domestic sheep. Other evaluations that were made were that all bighorn sheep died within 24 hours of showing clinical signs of the pneumonia, which reflected the rapid fatal nature of the pneumonia. Stress factors that were acknowledged in the paper were human manipulation, transport handling, incurrent disease, and environmental factors such as weather. These stress factors can often predispose the animals to pneumonia. Although the researchers included the stress factors, it was determined that none of these factors ended up playing a role in the death of the bighorn sheep. In conclusion, bighorn sheep and domestic sheep should not be managed in close proximity to each other because the bighorn sheep will likely contract pneumonia that is fatal.
After studying this research paper, we believe that the experiment was not done the best that it could have been. The research could be supported by having more than one trial and multiple sets of results to prove that the results they gathered were correct. We also thought that the paper would benefit from more visual aids. The one table that was provided was difficult to understand, and that is why we decided to include a supplementary graph. If this experiment were repeated, we would suggest that the researchers include more sheep, both bighorn and domestic, in the study. This is important because it will give a better idea of how the bighorn sheep are impacted by the domestic sheep. The research could also benefit from having domestic sheep from different origins and a wider age range available. Having separate herds is one way that multiple trials can be conducted. Having multiple trials is important in research to prove that the results you obtain can be repeated.
A possible future experiment could be that researchers can also use these results to try to develop a more effective vaccine for the pneumonia so that one day bighorn sheep and domestic sheep can commingle without fatal consequences. This would alleviate stress on the farmers in Oregon and the Pacific Northwest to have peace of mind about not endangering the bighorn sheep anymore due to P. hemolytica. The bighorn sheep play an important role in the ecosystem of this area due to their participation in the food chain.
The results of this research can lead to more experiments on other wild and domesticated species that could have possible contact. For example, wild boars often come in contact with livestock and humans. They carry many diseases such as swine fever, tuberculosis, and some zoonotic diseases. Using the design of this experiment to research other species and diseases can help to slow the spread of diseases between wild and domesticated animals and possibly even humans.
These are our references used throughout the presentation, including pictures and the original research paper.